Hello everyone, welcome to Violation. Now in this video we are going to continue with the second part of the chapter Genetics, some basic fundamentals. So let's begin. Now who is known as the father of genetics? We all know that Gregor Johann Mendel is known as the father of genetics, and he is known for his experiment on the pea plants, and the scientific name of the pea plant is Pisum sativum. Gregor Johann Mendel was an Austrian monk. He studied the inheritance of the traits in the pea plants, and he developed the laws of inheritance. And Mendel's work was not recognized until the turn of the 20th century. Now let's have a look on the Mendel's pea plant experiments. Why did Mendel select the pea plants for his experiment? Now there are some reasons for it. So the reasons are: the pea plants can be grown in a small area. The pea plants have 7 pairs of contrasting characters. The pea plants produce lots of offsprings. The pea plants produce pure plants when allowed to self-pollinate several generations. And the pea plant can also be artificially crossed. Now the 7 pairs of contrasting characters studied by Mendel are as follows. So the first character was seed shape. The dominant trait was smooth and the recessive trait was wrinkled. The second character was seed color. The dominant trait was yellow and the recessive trait was green. The third character studied by Mendel was pod shape. So the dominant trait was inflated and the recessive trait was wrinkled or constricted. The fourth character was pod color. So the dominant trait was green and the recessive trait was yellow. Next, the fifth character studied by Mendel was flower color. So the dominant trait was purple and the recessive trait was white. The sixth character studied by Mendel was flower position. So the dominant trait was axial and the recessive trait was terminal. And the seventh character was stem length or the plant height. The dominant trait was tall and the recessive trait was dwarfed. Now what did Mendel do? Out of the characters he selected one character at a time. Then he crossed the 2 traits of the character to observe the results. Mendel removed the stamens from the purple flower and then transferred the pollen from the stamens of the white flower to the carpel of the purple flower. Next, the pollinated carpel matured into pods. Mendel planted the seeds from the pod and got the F1 progeny. Next, Mendel's result. So when he crossed the 7 pairs of contrasting character, he got only 1 trait in the first filial generation. And he gave the term factor. Now we term it as gene. Our reasons for Mendel's success. Why did Mendel succeed? Mendel worked with only 1 character at a time. He carried out experiments to the 2nd and the 3rd generations. He built up sufficient amount of accurate data so that he could come to a scientific conclusion. He performed the crossing techniques with proper care during cross-fertilization, and he emasculated the flowers early to avoid self-pollination and bagged the flowers after pollination to avoid the deposition of pollens from some other plants. Now there are 2 types of genetic crosses: the monohybrid cross and the dihybrid cross. So monohybrid cross are the cross which involves a single trait at a time, for example flower color. And dihybrid cross are the cross which involves 2 traits at a time, example flower color, and plant height. Now, what are Punnett square? So, the Punnett square is a square diagram which are used to predict the genotypes of a particular cross or breeding experiments. Monohybrid cross. So, monohybrid cross are the cross which involves a single character. For a monohybrid cross, first ascertaining homozygosity or the pure line is essential, and the parental cross yields offspring of the first generation, which are called the F1 generation or the first filial generation. So let us take one example to explain the monohybrid cross. Here, pure tall pea plants were crossed with pure dwarfed pea plants in the first parental generation. So the genotypes of the tall pea plants were T, and the genotypes of the dwarf pea plants were t. Now from the tall pea plants, the gametes obtained were T, and from the dwarf pea plants, the gametes obtained were t. Therefore, the progenies obtained in the F1 generation were all T, that is, all the plants were tall. So the phenotype of all the plants were tall and the genotypes were T. Now by selfing of the F1 generation, we got, that is, T into T in the second parental generation. Hence, in the F2 generation, 3 plants were tall and 1 plant was dwarfed. Therefore, the phenotypic ratio were 3:1. And as there were 3 types of genotypes, that is 1 T, 2 T, and 1 t, the genotypic ratio were 1:2:1. Next, Mendel's first law of inheritance, that is the law of dominance. The law of dominance states that in heterozygous condition, out of the 2 contrasting alleles, one expresses itself morphologically and the other remain unexpressed. The allele which expresses itself phenotypically in the presence of its contrasting allele is called dominant, and the other, which remains unexpressed, is called recessive. Next, Mendel's second law of inheritance, that is the law of segregation. The law of segregation states that during the formation of the gametes, the 2 alleles responsible for a trait separate from each other.